Okay, great. Over to you, Benjamin.
Could you confirm that you see my slides as presentation mode?
Yes, we can see them.
Perfect. Thank you. So hello, everyone. I cannot see you, so I'm just talking to my computer, but I hope you can hear me. So I'm Benjamin. I'm a research group leader at the Center for Reproducible Science at the University of Zurich. And today I'm going to talk about how systematic reviews of animal studies can foster reproducible and translatable research. And so we are at the STRIDE lab from the Center of Reproducible Science. And the goal, so the overarching goal of our lab is to foster the development of therapies to treat human diseases. This is also called translation. And although translation is a different concept than reproducibility, it is somewhat related. It's related or it's basically generalizability from an animal model, for example, to a human population, which is just another kind of animal. And so this is somewhat closely related to the concept of reproducibility. And we work with evidence synthesis and data science, including systematic reviews. And like we aim to impact animal welfare. This is one of our main goals, what we hope that our research can achieve. And actually today, Valentin already mentioned that I'm a proxy for Mariana, who couldn't make it, but she sends her regards. And while I cannot replace her, I hope I still can give you an idea of systematic reviews and how they can foster reproducibility. And if you're interested, we have a lab web page which you can find. I assume the slides will be shared with the participants. Before I delve into systematic reviews, very briefly, I assume many of you know this, the 3R principles suggested in the late 50s by Russell and Birch. And so these 3R, that's replacement, reduction, and refinement. And these are somewhat concepts or principles how animal experimentation should be conducted, or with the mindset behind it. And the replacement aspect refers to replacing as many animals as possible with non-sentient types of experiments like in silico, computer simulations, or cell cultures. Reduction refers to the concept of using less animals, but still being able to draw the same conclusions, closely related to the talk we just heard before, like, for example, using more powerful statistical tests, for example, where you can reduce your animal counts and still draw the same conclusions. And finally, refinement, which is about reducing or minimizing the harm you impede on the experimental subjects, the animals. And so these 3R principles, they always have to be considered when I think many of you have experience with writing ethical applications for conducting animal research. And you always have to argue, how did you consider the 3R principles? And that's the blue arrow on the right side of this balance. So the harm you inflict on the animals, or the 3R to reduce this harm overall. And on the left side of the scale, you have the benefit. What will you potentially gain from these experiments? And you have to weigh these two things in this balance, and this is called the harm-benefit analysis. And you have to basically make your point that your experiment has a lot of benefit, while as minimal as possible harm. And systematic reviews, they basically can shift this harm-benefit analysis by reducing the harm, the overall harm. Like we're not talking about individual animals, but the overall conceptual harm you have on the animals, and they can increase the benefit of the overall conclusions you might be able to draw. But for those of you who don't know what a systematic review is, I actually haven't told you that. But a systematic review is some type of a literature summary, but it is not just, you don't just summarize literature, but it has a very rigorous and structured and thus reproducible process of methodology behind it, how you conduct the systematic review. And with this they can be quite impactful depending on how rigorous you conduct them. But it has been shown that these systematic reviews have been very important to uncover certain problems in preclinical research, like, for example, poor reporting or unrealistic experimental standards. With this, they have informed practice guidelines for animal research, for example. They can reduce research waste. For example, if you are able to show that certain experimental models don't translate well to a human population, which you should reconsider in such a case. They can guide translational research. And finally, and I think very importantly for this crowd and this seminar today, is they can enhance reproducibility directly and indirectly. And while I won't give a very detailed methodology of how you conduct a systematic review, very briefly, like for didactic reasons, we usually say it consists of 10 steps. And very briefly, you define a research question you're interested in. For example, does drug A enhance functional recovery after stroke in a rat model of stroke? And then you have different expertises. You might have statisticians who help you with the systematic review process, the meta-analysis, for example. Then you might have information specialists who help you with the search, and then you have field experts in the field of stroke, for example, who know the field well. And then you search for pertinent literature in different databases. So, for example, PubMed or Embase, or there's lots of such databases. Then you write and register a study protocol in which you define which methodology are you going to use in the systematic review? What are the criteria for including articles, and what is your research question exactly? That's a relatively long document you should a priori register. And then, based on your search, you basically sift through all the studies you found and check, are they relevant to answer my question, yes or no? And you extract data from these articles to potentially inform your question. Like you manually extract, for example, certain numbers from graphs or from the text. And then you assess the risk of bias. This is somewhat related to study quality. It's not exactly the same, but just that you have an idea what this is. And then you basically merge together all these data and draw a conclusion on your initially posed research question. And in the following slides, I would like to show you how systematic reviews can foster reproducible research, and for particularly translation from animals to humans, how they can inform the better translation. And this is a master thesis from Ingrid. She's a medical student, master student, and she has basically looked at multiple sclerosis drug development. Multiple sclerosis is a chronic neurological disease. It affects mostly young adults, and it's a chronic disease, so you have it for the rest of your life if you get it, or if you contract it. And it typically presents with these white patches in the brain MRI, as you can see here. And we were wondering, so multiple sclerosis is actually quite well treatable nowadays. So there is more than a dozen approved therapies available to treat multiple sclerosis. And over the last 20 years, these have obtained regulatory approval and are nowadays used in the clinics to treat patients. And at the same time, there is also drugs which were tested in patients, in MS patients, but maybe they were toxic, or that they didn't show an efficacy, so they dropped out during the clinical development, and so they're not used today. They failed in clinical development. And so we asked the question, could we have predicted whether a drug obtains regulatory approval or whether it would fail in a later clinical trial based on the animal experiments? So could we have known in advance which status a drug will achieve later based on the outcomes of the animal experiments? And that's what Ingrid basically done in her master's thesis. And so this is a systematic review, obviously. So I won't go into the detail of the methodology, but I can say we have included around 500 articles relevant to answer this question. So these are animal studies, 500 animal studies, which tested drugs in multiple sclerosis animal models, and approved drugs and drugs which failed later in clinical trials. And we found a couple of quite interesting things, I would say. So first, we looked at all these studies. We looked at what types of animals have been used by these studies, what type of animal models have, what sexes, male or female animals have been used, and which strains, and a couple of other things which I'm not showing here. But what is striking about this graph is that there's an extreme homogeneity how these drugs have been tested. So in summary, 80% of the drugs have been tested in mice with one animal model, the EAE. This is the experimental autoimmune encephalomyelitis. This is obviously a commonly used animal model for MS. And over 80% of the studies used female animals only, exclusively. And also for the mice, one particular strain, the Black 6 mice, have been used in the large majority of these studies. So this shows that there's an extreme standardization of testing procedures for the drugs, which might be a problem if you generalize it to a human population, which is very heterogeneous. The second thing we looked at is the effect sizes. So one thing we hypothesized, or we thought could be true, is maybe drugs which are nowadays used in clinical practice, maybe these drugs have stronger effects in the animals compared to drugs which failed later. So that was our hypothesis. And to look at that, we particularly looked at this EAE score, the clinical score you can see here. You basically measure how well the mouse, or how badly the mouse, or how functionally impaired a mouse is from 0 to 5. 0 means the mouse is completely healthy, and 5 means the mouse is actually it's dead. And so from 0 to 5, and we looked at the effect sizes, and again we would assume that drugs which reach regulatory approval would have stronger effect sizes. So the drug used to treat these mice would lead to a better recovery of the mice, maybe from 4 to 0, or from 5 to not from 5, but from 3 to 0, and the drugs which failed later would show smaller effect sizes. However, this was not what we saw when we looked at these effect sizes. So to explain you this graph, in blue, you have the drugs which are nowadays used in clinical practice, so the approved drugs, and in red, you have the drugs which failed in clinical trials. And at the bottom, you have this black diamond, so this is a forest plot basically, and on the y-axis, you have the effect size. And important to acknowledge in this graph is that these two black pooled effect sizes for approved and failed drugs are exactly the same, basically. So both these drugs show the same effect sizes in the mice. So our hypothesis was incorrect. And the third and final thing we looked at in this study is also quite striking, I would say. It's probably the most striking thing from this study. So we also looked at the timeline of these drugs. So on the y-axis of these graphs, you see the timelines on when a particular study, an animal study, has been conducted. So every single gray circle is one single animal study, or basically the publication of that animal study. And then you also have orange circles, which correspond to regulatory approval of the drug. So in this year, the drug obtained regulatory approval to treat patients. And the green circles are the years in which the first in-human trial of this drug has been conducted. And on the left side of the graph, you have all the approved drugs, and on the right side, you have the failed drugs. And I'm sure you have already noticed, but I still want to point out that if you look at these numbers, 90% of all the animal experiments have been conducted after the drug has already been approved for treatment, and after the drug has already entered a clinical trial for humans, which is quite surprising, I have to say. I'm not saying that there is no reason to conduct animal experiments for testing a drug, even if it obtained regulatory approval. However, there seems to be a disconnect between animal research and clinical research regarding the reasoning behind conducting animal experiments. I would claim, I don't have evidence for that, but I would boldly claim that some or maybe many of these animal experiments would not have been necessary because the drug is already used in a clinical setting. So basically, to summarize this project, it shows you that by looking at systematically at the literature on a certain topic or on a certain research question, you can unveil problems in preclinical research which could help to foster reproducibility in animal research. And in the following, I have two more projects I would like to briefly show you. And this is more, the next project I'm going to talk is more on a meta aspect of systematic reviews, which we're also interested in. And this has been the master's thesis of Julia, another medical master student in our lab. And she has basically looked at how long does it take for researchers to conduct a systematic review. So we are interested in fostering this methodology because we are strong believers in it. So we also have to tell people or give them some estimate, okay, how long do I need to conduct such a systematic review? And I think these are important things to know for a researcher before embarking on such a project, because sometimes people might have unrealistic expectations. And so how did we do that? We took a heuristic approach. Basically, what we did is there is this called Prospero. Maybe some of you have heard about that. This is a registry. It's like, yeah, it's a registry, an online registry where you publish I mentioned before, this systematic review protocol, where you define your methodology in advance before you conduct a systematic review. And if you conduct a rigorous systematic review, you have to upload this protocol to this Prospero database. There is other databases, but this is one of the most important ones. And what we have done is we looked at all these protocols and checked how many of these protocols actually resulted in a publication. So how many of these reviews were completed and published, and how long did it take from the protocol registration, in which the authors actually also have to claim when the review starts. So this gives us the relatively exact start point of the review until it has been published or submitted to a journal. And we assume that the submission to a journal equals roughly to the completion of the systematic review. And we downloaded around 1,800 protocols related to animal studies. So these are only animal systematic reviews from this Prospero database. And we had to exclude some due to a couple of reasons I won't go into now. But one of the first main finding was, you see here in the middle of the figure, 724 of these protocols have resulted in publication. By the way, how did you check that? We went on different databases and searched for a combination of author names and titles, keywords, and try to figure out the link between the protocol and the publication. And the first main message of this project is only around half of or a bit less than half of the protocols are actually completed and culminate in a publication. So less than 50%. We also looked, it was fun to do. And I think it's also quite interesting. We looked at which countries are most prolific in publishing these systematic review protocols. And quite surprisingly, I didn't expect that, but Brazil is number one. Brazil is the most prolific country regarding the systematic review protocols. We didn't normalize this data to the number of researchers. Obviously, this is just the absolute number, but I think it still gives some idea on the global contribution to these protocols. And finally, to answer the main question of the study, how long does it now take to complete a systematic review? Well, this is didactically not very good. From me, I should have shown you all the thing, but basically the author said, so they also have to, in the protocol, they have to say how long does it take to complete such a review. And they say on median, it takes 7 months. However, it takes around 11 months to complete such a review. To complete it, to submit it to a journal, and then the publication process basically takes another 4 months, which is actually quite a long time, but this is how the system works. So if you plan to conduct a systematic review, this gives you some concrete number how much you can expect the amount of required time you need. Obviously, there is cases where you can complete a systematic review in 1 month, or there is also systematic reviews which take 2 or 3 years. So that's not the guarantee, obviously, but gives you some guidance. And to finalize this talk, I would briefly like to dig into another problem which we're working on. Is every one of you in this lecture hall is aware of this problem, that the number of publications is just exploding. No one can read that. It's impossible. And all these data also are in a very unstructured format, so they're just buried or hidden in publications in a relatively unstandardized fashion. So we are interested in actually automating this whole process of systematic review, or let's say semi-automating. There will, or in my opinion, there will always be some human in the loop. And this is obviously a very complex process, but we are interested in at least certain steps of the review process, like the abstract screening or the data extraction could be at least semi-automated. And a PhD student of mine, Simona Doneva, is working on that. She's a computer scientist, and she uses large language models like GPT, or there is other models actually which are very good and open source. And she's basically training these models to extract information from clinical texts. In that case, it's clinical trial registry texts, but this could be extrapolated to other types of texts like scientific abstracts. And without going into details, these models show a human-like performance if you train them well. So, and I want to emphasize, we're not just asking ChatGPT. This is like using the underlying model and training it to do a very specific task. And these models show human-like performance for even complex extraction tasks. And so the goal is, why do we want to do this? Where we want to go is, we want to actually build a data warehouse. As I mentioned, we're interested in translation, so in drug development from animals to humans. And we want to mine every single publication which has tested a drug in an animal model of a neurological disease, and want to summarize this all in this data warehouse. And I brought this, we have a pilot, we called it Anemone, Animal Models in Neuroscience. And the idea is that a researcher goes to this database, and they could check how many animals have been used in total for all these experiments, what is the overall quality. Then you can select therapies you're interested in, then you can see in which kind of species and models have they been tested, have they been translated to humans, what was the outcome. You could also, for example, select only therapies which have not yet entered any clinical trial. Then again, you could check how many animals have been used in this setting. And then you could also go to the data warehouse where each individual study, so this will be hundreds of thousands of studies, is listed per row, and you have metadata linked to the full text of the study, then which animal has been used in the study, how many animals have been used, which therapy has been tested, what was the outcome of the study, and what is the overall quality of these studies, and whether it has been translated or not. And this is work in progress, but I still wanted to share this pilot data warehouse with you. And so what to remember from this talk? I think systematic review is a very a methodology which can help you in your research. For example, to assess translational hurdles of preclinical research, to benefit animal welfare. They can be conducted within a reasonable time frame, roughly 11 months. They can potentially be semi-automated to even make them more efficient and to even make them more easier for you to use. And finally, they can foster reproducible research. And before I stop, I would still like to do a small advertisement. So if you are now, after this talk, or even before that, are interested in systematic reviews, you can get in touch. So we are the Camarades Group Switzerland, actually. So we are a a group of people who help animal researchers in Switzerland and beyond to conduct systematic reviews. So if you want to conduct one or are in process, we're happy to support you methodologically. And if you want to dig deeper, we are also conducting a summer school in Bergen in Oslo, which is very beautiful in late summer in August this year. You can apply. It costs roughly 300 euro, which is relatively cheap, and you will learn basically how to independently conduct an animal systematic review, which we believe is very important as a goal of this course. And with this, I have talked enough. I thank obviously my team. They've done all the work, our funding, and I thank you for your attention and happy to take any questions.
Okay, thanks, Benjamin. Yeah, let's see if we have a question in the room. Any question online? Okay, there's one question here. Yeah.
So on your slide 8, that was the one about animal tests being after FDA approval and yeah, exactly, and the first in human trial. I mean, this, of course, this is not causative leading to the first in human trial. It's simply when the drug's around and it's associated with a disease, then it will be used in other studies regarding this disease, and it can also go down the line of drug repurposing, I assume. I mean, this is probably am I too naive looking at it like that? But that's why this happened.
But can I ask you back? Would you expect still that 90% of all the animal experiments testing a certain drug are conducted after the drug is already used in the clinic? I mean, or let me add this. I agree. As I said, there is reasons for conducting drug testing after official regulatory approval. You mentioned drug repurposing, which is correct, or mechanistic investigations, but still 90% is still quite a big volume. Or did I misunderstand you? Maybe.
No. I wouldn't have put a number on it. But the thing is that the name of the drug's not around until it is approved or used. So it can only appear in publications after it's there. So
okay, that's a good point. Yeah, we actually included all synonyms, including drug codes. Like, for example, if a drug is under investigation by some pharma company, it has some GSK 55564, and we have a comprehensive list of synonyms also included in this analysis. So this should not be a problem.
But maybe it's again also publication bias. I'm not sure, for example, if the pharma companies really publish so much of the animal studies when they actually line up their phase 1 study and so on, right?
It is a valid point. There is certainly publication bias, and in addition, publications or studies tended to be less published maybe in the 90s or in the 80s. So there's other factors driving this. I'm not saying all of these 90% of the animal experiments are completely wrong. I'm just saying this shows potentially a bigger problem, a potential disconnect, even though there might be some mitigating factors here, but still the 90% is just a lot.
Thank you for your talk. When you say first in human trial, is that actually in a patient population, or would that also be in a healthy volunteer PK study? So if that is the case, then obviously this study would be conducted fairly early, I assume. And then obviously there would be an overlap. I mean, 90%, I agree it's a long time, but if you also refer to healthy volunteer studies, then obviously these are conducted early on.
Good point, but these are actually the first in MS trials. I didn't specify it. That's a good point you're raising, but this is not phase 1. This is, I mean, potentially phase 1, but these are just MS patients or persons with MS, not healthy individuals.
Okay, thank you for clarifying.
Yeah, thanks for asking.
Okay, I think we have to move on. Thanks again, Benjamin.
Thank you.
Great presentation. Thanks for jumping in. And